Our next author, Dr. Bleep. We had them quite a while ago. 2546. I think that's like that was like 400 SCPs. Jesus. 2546. It's exactly 400 SCPs ago, actually. And it's the I think it's the it's the two clone. Well, is it two or is it multiple? Well, it's basically like you're looking at a clone of yourself, and one of those clones will try to kill you. yeah. A retroviral nightmare. Yep, that's what it was. That was man, 400 SCPs ago, dude. That's crazy. It's been out since September. September, like, so it hasn't been out for long, but it feels like forever ago. Alright, Dr. Bleep, we got overly large E. coli. Okay. Okay now. Upper class Euclid. Why are you making them look like the sour worms? You know what I mean? Like these guys. You know what I mean? Don't they look like that? Like even like the color right here. It's like this one. And this one? No, okay, there's no color that fits it, but like, yeah, it just looks like the sour worms. Upper class Euclid. Special containment procedures. SB2946 is to be kept in a 125 cubic meter containment tank with light, durable metal edging and plate glass sides. Well, a colorful picture, it is colorful. The bottom should be constructed with 5 drainage channels, which are to remain sealed until completion of protein expression. These drains will be connected to a protein vat, 4 sterile tanks capable of holding 200,000 liters of broth are to be attached to the tank via sterile PVC pipes. Pipes should be more than 15 cms in diameter and should have both manual and mechanical control valves. Broth tanks shall be filled with nutrient broth composed of the following ingredients: 2% peptone, 0.5% yeast extract, 100 NaCl, not sure what that means, 25 MnKCl chemicals, 150 MgSO4, and also 150 MgCl2, 200 glucose, and antibiotics as needed. Me and Dad eat those SB gummy worms, by the way, Afro. Of course we do. SB2946 specimens at the bottom of the bile, salt broth pool where they were discovered. On the east and west sides of the tank, respectively, a cell sorter and large buffer basin should be constructed and attachable with appropriately sized PVC pipes. The tank shall be placed on top of a large-scale shaking mechanism and rotated at a constant 135 RPM. Temperature should be kept at a constant 37 degrees Celsius until the end of each 5 replication cycle period. Glucose concentration are to be monitored regularly. If concentration fall below predetermined levels, glucose is to be added until they reach acceptable standards. Every 5 replication cycles, glucose concentration are allowed to fall to 0. The tank is to be sealed immediately. Temperatures lower to 30 degrees Celsius, shaker set to 300 RPM and 100 mm IPTG added. The tank is to remain in this state for the next 4 days. At the end of 4 days, all broth is to be drained from the tank. The tank is to be flushed with phosphate buffer. The cell sorter is attached and all but 8 individuals removed from the tank. Researchers working with or around SB2946 is in non-transformation periods are to be observed BSL-2 standard protocols. In the event of a containment breach, trapped personnel are to be submerged repeatedly in a tank of lysozyme EDTA in hypotonic conditions. An enzyme commonly found in human tears and egg yolk which degrades the cell wall of bacteria. During transformations procedures, all personnel are to wear BSL-3 standard protective gear in the event that the entity is exposed to any foreign DNA other than designated plasmid, a level 3 purchase to be initiated, and a containment tank flooded with concentrated hydrochloric acid. Research requests for exposing SCP-2946 to foreign genetics materials require level 4 approval. Show extended explanation for containment procedures. On a normal growth in conditions, SCP-2946 can reach density of roughly 3.4 times 10 to the power of 5 cells per 125,000 liters of broth after 5 doubling periods. When cells reach this density within the tank, they become entangled and immobilized by the surrounding cells, minimizing the chance of a breach event or self-rupture. It is at this point that glucose input is reduced to 0 and concentration allowed to fall all the way to 0. Absence of glucose subsequently facilitates the activation of the plasmid encoded lac promoter, initiating production of the desired protein. The lac operon is one of the commonly used examples of metabolic regulation. In the absence of glucose and presence of lactose, allolactose will bind to a repressor protein on the top of the promoter and cause it to release, allowing transcription to occur. And then to further amplify expression, IPTG is added in large concentration. It's all, by the way, still the containment procedure. We still don't know what it does. It's all still containment procedure. Isopropyl beta D1 thiogalactopyronoside, a molecule which mimics the structure of lactose. Due to the volume of the containment tank, 3 to 6 days of the conditions described in the containment procedure are required to accumulate appreciable protein quantities for analysis. Once all broth has been drained from the containment tank, the previously described basin containing phosphate buffer and a gelatin agent are attached to the tank. SCP-2946 cells will attach to surfaces when exposed to dry air and can be difficult to remove. Use of gelatin agent such as ETTA removes the protein receptors and weak force interactions, facilitating this attachment. Furthermore, once they've been detached from the surface, specimens are sorted via a large-scale cell sorter based on the intensity of the fluorescence and the size and number of cellular structures. Individuals that fluoresce more intensely and have numerous cellular structures are considered more fit for continued culture. 8 of these specimens are returned to the tank, while the rest are removed and ruptured for internal analysis and nutrient recycling. The remaining 8 individuals are prepped for a standard bacterial transformation and inoculated with a fresh plasmid containing a different resistance marker from the previous entry. SCP-2946 is not to be combined with SCP-3536. Well, that's in the far future. Well, not really far future, but you know what I mean. In the future. Ooh, booyah. Booyah, I tell you. Description. Finally, the description. SCP-2946 is a strain of Escherichia coli with a radius of 10 centimeters and a length of 40 centimeters. I also see how many footnotes this one has. God damn. Thus a volume of roughly 2,100 centimeters cubed. In both nutrient broth and dry air, individual cells have a large range of pigmentation and actively fluoresce. An example, emit color when struck with specific wavelengths of light. Individual cells may possess 1 or 2 flagella, allowing them to swim through the tank in less dense conditions. A common protein structure used by bacteria for movement. Specimens display impressive leaping capabilities, able to propel themselves from a liquid environment at speeds of near 100 km per hour to heights of 30 meters or more with the use of these flagella. SCP-2946 doubles once every 2 weeks, with the 2 subsequent specimens expressing different pigmentation and fluorescence from the original. Each cell appears to defy certain biological principles derived from the square-cube law due to its relatively large size and should not be able to metabolically sustain themselves, much less replicate every 2 weeks. It says that as shape grows in size, its volume grows faster than its surface area. In biological application where cells depend on the surface areas for nutrient transports, this means the larger the cell, the more inefficiently it metabolizes and transports nutrients. Furthermore, when exposed to certain conditions, metabolic rate, protein synthesis, and replication increase at an exponential rate, often resulting in lysis. Despite identical membrane composition to the smaller cousins, specimens are able to diffuse and actively transport nutrients for far more efficiently, transporting glucose from the surrounding broth at a rate of 36 mg per hour. Inexplicably, when exposed to lactose in the absence of glucose, SCP-2946 quadruplets its rate of protein produced. In some instances, the cells may even rupture or perish due to extreme overexpression of toxic or large proteins. SCP-2946 possesses cellular structures normally only found in animal, plant, or fungal cells. These features allow them to produce proteins which are normally non-functional in bacteria. Additionally, due to the presence of certain structures, individuals are able to secrete protein into the surrounding broth, making collection and purification of proteins via lysis of individual cells unnecessary. A relatively simple but often time-consuming process. This makes SCP-2946 an invaluable tool for research purposes, enabling a collection of large amount of protein within a short period of time, using relatively few resources in the long term. Most individual cells are relatively harmless during a normal growth states. When deprived of glucose, entities will move towards a nearest source, including humans, utilizing glucose sensors in their flagella. Microscopic structures which behave similar to dowsing rods pointing the organism in the direction of glucose. I'm just wondering, is this like how actual E. coli acts as well? Because I don't know, I'm not very like versed in the behavior of E. coli. Upon reaching a source, SCP-2946 will begin secreting corrosive fluid to break down tissue and acquire glucose. An array of digestive enzymes and exotoxins not found within the cell's genome. Strangely, the organism is not immune to its own corrosive secretions, often rupturing itself long before it digests the target source. Rate of digestion depends on the number of active individuals secreting onto a subject. A single specimen is incapable of digesting a human host alone and will perish if it engages in prolonged excretion. 5 or more individuals are capable of digesting a human source in as little as 30 minutes, unable to survive due to the reduced exposure time. In most cases, consciousness in the human host is not lost until the subject expires. God damn. Just eating a whole human in 30 minutes? God damn, boy. Single cells carry several million copies of a large single circular genome and may carry tens of millions of copies of one or more plasmids, depending on the current line of experimentation. SCP-2946 is extremely susceptible to foreign genetic material when undergoing transformation procedures. Unlike its smaller cousin, non-plasmid DNA taken up by the cells is immediately integrated into its genome. Uptake of animal, plant, fungal, and even protist DNA have drastic and poorly understood effects on the psychological characteristics of impacted cells. Notably, when one group of cells was accidentally exposed to... I need to ask him, actually. He's not awake by now. He's probably dead asleep, but I need to ask. Okay. It's extremely unlike its other small cousin, non-plasmid. Uptake of animal, plant, fungal, and even protist DNA have drastic and poorly understood effects in the physiological characteristics of impacted cells. Notably, when one group of cells was accidentally exposed to a strand of hair from ductum, effects were immediate. All cells lost their rod shape, became circular, and began dividing rapidly, clinging to each other until individual cells were indistinguishable. The mass of expanding cells formed large-scale organs and limbs, including a heart, 2 lungs, what appeared to be a hair follicle cells, and were ducted. Before the tank was sealed, junior researcher was swallowed into the tank by one of the flaying limbs. The cells swarmed her shortly thereafter, whereupon visual contact was lost. Upon being sealed, the tank was flooded with hydrochloric acid. I love how that's your first question. After all this, it's like, so it's an E. coli? I'm like, that's what it literally says. That's what the title even says. Following this incident, current containment protocols were put in place. Inquiries into further effects of foreign genetic material on susceptible individuals are now required to undergo level 4 review and approval. See experiment log E13 and extended collaborative experiment log E34. Discovery. SCP-2946 was discovered on August 13, 1994 by a group of Foundation researchers exploring SCP-2378. The organism was first observed leaping from... I was zoning out for a bit, okay? Okay, sure. You say that as you said before. Oh, I'm liking this SCP. What is it? The organism was first observed leaping from a pool on bile salt towards a large stalactite from the ceiling, missing several times before hitting and breaking the structure off and into the pool, where it immediately dissolved. Later determined to be composed of glucose, lactose, and mannose. Intrigued, researchers set up recording and measurement equipment and spent time collecting environmental samples, pH measurements, and observations. At approximately 14 hours, the researchers broke for lunch, and Dr. was seen consuming several very sugary foods. At 14:10, Dr. was swarmed from behind by approximately 30 instances of the entity and dragged into the bile salt pool. The remaining members of the research team immediately called for a containment crew and made efforts to fish Dr. from the pool using collection nets before it disappeared from visible view. Can you imagine, like, you turn around and there's this bacteria the size of a human being and it just like grabs you and just like and like, no! Containment crews arrived at 14:20 and were briefed on the situation. Individual cells were captured without incident once the pool was drained. Dr. remains could not be located. All instances of SCP-2946 and more than 100 kilograms stalactites were collected by both the research team and containment crew for study and appeasement purposes. Level 2 clearance required. Experiment log E. I also love how it's like E something, right? Because E. coli. Yeah. E13. All researchers requesting and conducting tests with SCP-2946 are to record the results in the format below. Our experiments were conducted in a biosafety level 4 chamber and flooded with hydrochloric acid immediately after tests were concluded. Test 1. August 30th, 2000. Dr. Andrews. And then the Metacyclostrata tropical fern. Okay, tropical fern. Okay, so here we have the result. So I would say 30 seconds. Cell pigmentation changes from purplish tone to green. Cell shape shifts to a red, rod-shaped cylinder into a rigid polygon with a thick cellular wall composed of cellulose. Light absorption measurement within the chambers changed, indicating increased reflection of green light and increased absorption of red and blue light. Organelles typical of plant cells promptly appear, included chloroplasts. 1 minute. Cells begin rapidly dividing, clinging together and differentiating into characteristic plant tissues. 5 minutes. Large leaves characteristic of ferns sprout and begin filling the room. 6 minute leaves develop razor sharp edges and begin flailing against the observation windows. 10 minutes. Structure analogous to Dionea muscipola manifests and begin secreting highly corrosive fluid into the window, colloquially known as the Venus flytrap. Chamber is flooded with hydrochloric acid. Date September 3rd. Researcher Dr. Andrews. Sample identity Amanita muscaria, red and white spotted mushroom. 30 seconds. SCP-2946 progresses from a rod-shaped cylinder into a long filament with multiple nuclei. 1 minute. Filament divides rapidly into branching / interweaving structures, forming hyphae and subsequently mycelium? I don't know. A lot of scientific terms being thrown around. 2 minutes, 16 large frilled red-capped toadstool grow from the network of filaments. Aerial spores are detected within the chamber. 5 minutes, structures analogous to roundworm oral cavity burst from the cycilium and begin flailing spasmically against the walls and observation windows. 7 minutes. Toadstools begin secreting aerosolized psychoactive toxins similar to those found in ergots. 10 minutes. Filaments condensed beneath toadstools' prehensile limbs with claw digits begin forming a pairs of tens. Rumors flood with hydrochloric acid. Date September 5th. Dr. Andrews again. Acrosis, roseas, rose, slime, mold. 30 seconds. Cell loses all rigidity, becoming amoeboid. 1 minute. Cell begins rapidly dividing, spawning thousands of additional amoebae. 2 minutes. Cells begin aggregating and form a characteristic fruiting body structure. 5 minutes. Structure begins rocking back and forth wildly. Long tentacle-like structures begin extending from the base. 6 minutes. Each tentacle develops hundreds of openings with sharp teeth-like protrusions. 7 minutes. Top of fruiting body spawns large spherical structure. 8 minutes. Spherical structures drop into dentata of the openings. 9 minutes. Additional fruiting bodies grow from the spheres. 10 minutes. Tentacles begin to flail wildly, fruiting bodies sprout long serpentine limbs with 10 digits ending in numerous barbs. Rumors flood with hydrochloric acid. Damn. See, if you don't take E. coli seriously, this hopefully helps a little bit, because it's supernaturally doing crazy stuff is one thing. But in real life, and he just laughs at, yeah, of course. He's immune to all diseases and all bacteria. I think I need to stretch. Yeah, probably will do that real quick.